a crust, you can just chip it off with a forceps or a tweezer, and you can directly add it to a paper packet and collect it directly, send it directly to the FSL. The weapon directly can be sent to the FSL, because if there is a weapon found at the crime scene, there has to be additional physical testing of that weapon also, right? So in addition to biological examination, it will be subjected to physical examination as well. So you just collect the weapon as it is and send it directly to the lab. If the blood stain is dry, if there is a tissue or organ or a bone, it is to be placed in an airtight container after clean the bone. You're supposed to clean the bone of any tissue remaining, so that during the transport or the storage, there is no fungal growth on the bone, and it does not inhibit the subsequent creation of the DNA profile, right? It is to be stored in a transparent airtight container, sealed properly, kept in a refrigerator in case of tissue, and properly sealed and labeled and sent to the lab. In case of hair, obviously with root or with tissue or without it, you're supposed to collect it. Use the druggist wrap. I'll show you the druggist wrap paper. You're supposed to fold it 4 times, 6 times to prevent loss of such small micro evidences, and then place it into a paper envelope and further send it to the lab. How do we package it? We have collected the evidences. We have air dried it. Now our next aim is to send it safely to the lab for the proper analysis. So we use this kind of paper envelope which has all the details of the case. What are the contents of that envelope? What are the details? What are the nature of the evidences? How are you packaging it? In what? Suppose if you're packaging it in a transparent plastic container which has a red colored cap, so you're supposed to specify it here that this is what I'm putting the evidence in, and I'm sealing it with this much seals, and this is the person I'm authorizing to carry this evidence from the crime scene or from the police station to the forensic lab, so that any unauthorized person does not have the access to this evidence and destroy the chain of custody. In case of bones and tissues, we use such transparent plastic containers or transparent airtight plastic containers so that there is no spillage or leakage. Remember, in case of forensic analysis or for the DNA examination, you're always supposed to preserve the organs or tissues in normal saline. Do not use formalin. You're not supposed to use formalin. There are a lot of doctors who send tissues or abortus fetus in formalin, and that inhibits or that destroys the DNA completely. There will be no profile. You're always supposed to store it in normal saline and deliver it to the FSL as soon as possible, right? For evidences like nail clippings and hair samples, you're supposed to use such transparent plastic bags. And these will then, you're supposed to place it in a paper with the druggist fold, and then place that druggist fold inside this sealed paper envelope. This is the druggist fold. You place the sample here, you fold it from one side, then you fold it from the other side, and then you do another fold and place it inside the envelope. So this way, the small evidence that is present will not be spilling out, or there would not be loss of evidence during transportation or packaging. So this will be safe. Obviously, the problems that we face is if you're not air drying it properly, and if you're sealing wet samples directly, then there will be, fungal growth. Anything which is wet and sealed and does not have air ventilation is going to catch fungal growth. And once there is fungal growth on evidentiary samples, there will be no DNA profile, and a suspect or an offender will run free. Right? Do not force any packaging material into something that does not fit. It is going to break the evidence, or it is going to, deform the evidence if it is such in nature, and then that would not be helpful for solving the case. Depending on the nature of the evidence, you are supposed to use appropriate packaging material. Appropriate packaging material means if it is blood or semen or saliva or something that is wet, you're supposed to use paper envelope. If it is bone or tissue, you're supposed to use airtight bags. And if it is hair, then you are supposed to use Ziploc bags with the druggers fold. So you have to know what kind of evidence should be packaged in what way so that it will reach safely to the lab. You're always supposed to bring the container to the evidence and not take the evidence from the crime scene and roam about to a place where the containers are kept. Because during the transport, the evidence might spill or fall from your hand and there can be contamination. So always make sure that you take the container from your crime scene van and take it to the evidence. Each item should be packed and sealed and labeled properly as soon as you are collecting it, because if a crime scene has more than say 50, 60 samples and you're not documenting the nature of the place or you're not labeling it, when you reach your crime scene van, there are chances that you might forget the exact place from where you have collected it, or where, or the exact nature of the evidence, or exact person to which that evidence belongs, right? So you have to label it immediately after you have collected it. These samples or these packages that are created of the evidences should be clearly packaged with seals, right? If you feel that the places or the place where you have tied or where you have sealed these packages is, will not hold or will not maintain the chain of custody properly. If you're just putting one seals, then you are free to put in more, 2 to 3 seals or whatever number of seals wherever there are chances of easily opening the parcel or foul play happening. So you're supposed to, there is no guideline for a minimum number or maximum number of seals that can be used. You can put n number of seals as long as your evidence parcel is safe. So, what should be prevented while you're packaging? You are supposed to prevent contamination. You're supposed to prevent the loss of evidence. You're supposed to prevent foul play. Somebody changing the samples, somebody changing the evidentiary samples, planting evidences into a case. You're supposed to avoid that. You're supposed to prevent loss of chain of custody. Chain of custody has to be maintained at all times, because if at one step the chain of custody is broken, the entire case goes bad. So this is very important. It is indispensable to maintain the chain of custody. You have to prevent loss of evidentiary value of that evidence, which means you have to pack and label and store it so that the evidentiary value does not go down. Package parcel is to be properly labeled, and the package material, as I told you, needs to be appropriately decided upon. So this is the case. This is, suppose, an envelope which is storing the evidence that you have collected. So there has to be the case number of that case, the FIR number or whatever number that the police or the investigating agency is giving. The item number. Suppose if the evidence that you're packaging in this is 17th from the crime scene. So there has to be the number of that evidence. Name of the victim should be there. It depends. Obviously, you can avoid to mention the victim's name, but it's preferable if you write it over there if it is not such a sensational case. And the description of the item. Whatever you are packaging inside this envelope, you have to write it on the top of it. Say you're sending a plastic container with a blue color cap, which contains one EDTA vial with purple color cap, which is labeled as this. So you have to specify it on the top of this parcel so that the receiving agency can cross-verify with the custody sheet, chain of custody sheet, and then after the cross-verification, they can open this evidence box and the evidentiary value would remain intact. You have to apply tape on all the places where the joints are present, and initials of the authority and the date of the sealing. This has to be mentioned like this. And then at places like this, you have to place 3 seals here and 2 seals here, so that these are the areas where one can open this envelope and change the evidences. So you have to put 3 seals on this flap and 2 seals here, right? Packaging of the evidence to be mentioned to avoid any foul play. Main thing is you're to avoid and detect any foul play that is happening. So after packaging, next is sealing. This is an improper sealing because anybody can put their hand inside this envelope and bring out the evidence and can change the evidence at any place. This also, again, the top is open. Anybody can change whatever evidence is present inside. Uh, this is also improper sealing because anybody can open this knot and anyone can change the box that is there. This also is an improper sealed because this seal is not proper. Anybody can open it, change it. Just a flip needs to be raised, small flip of it, and then anybody can change whatever is happening, right? The labeling part. So this is how this parcel has to be labeled. You have to address it to the director of the investigating agency. You have to write the subject for DNA testing. There has to be the name, son of or daughter of, uh, whatever the age and the sex of that person is, the resident, the FIR number on the date of the FIR, and for DNA testing. It contains blood sample, EDTA plus red vial, one seal of SKJ. So this is a very proper labeling of an evidence because it is specifying whatever exactly is there inside the envelope, what are the details of the case, and if by chance after opening this envelope, the investigator sees that there has been a change in the EDTA vial or the labeling of the EDTA vial is different, he or she can identify that, okay, the doctor has by some due to some reason he has not put in the correct EDTA vial here, and then he or she can immediately report it to the police and then the remedial measures can be taken. So this is the internal number, the DNA number. I've blurred it so because you're not supposed to share the details, and this is the authorized authority's signature and stamp. Usually the IO sends it, but an SHO or SSP, whoever you think, or whoever, according to the police of your state, describes the details or the protocols, they can sign it and send it to the lab. This is the proper way of sealing. All the places where there are chances of the envelope opening has been sealed properly, and there has to be a sample seal along with this on a small cloth paper, a cloth piece, signed and stamped by the authority who has sealed this, so that while opening the parcel, the scientists at the lab can compare the seal and then ultimately open it. So this is a few example of the abortus fetuses that is there. And on the right side, you will see vaginal swabs or swabs that were sealed in the wet condition that has fungal growth here. So this evidence has no value. No DNA profile is going to be generated, or even if we get a profile, it is going to be a partial profile, which will not help you in giving any confirmative report. Chain of custody, I've told you the signature of the person who is sending, the name of the person who is the messenger or who is in custody of the evidence, the date of receipt and transfer, the date, description of the evidence, the marking of the evidence, and everything needs to be specified in that chain of custody sheet. If it is going from the IO to the SSL via a particular constable, that sheet is supposed to be written from the IO, authorizing that constable with his name and belt number and date in which he is carrying that evidence, with the details of the evidence, the number of parcel he has been handed over, the copy of the FIR, and a road certificate which specifies that he has started from the police station for the FSL on which date, and till what date does he have the authority to possess those particular evidences in his custody, right? So this is the forwarding of the case. There is a forwarding note while you're forwarding the evidences from the police station to the FSL. The forwarding authority details need to be present, the IO or the SSP. Usually the SSP or ACP, ADCPs are responsible for forwarding this. So their name and their region, their district of jurisdiction, the details of the case, the FIR or DDR number, the date of the FIR, the nature of the crime, the sections under which the person who is accused in that case, that has to be mentioned, the number of parcels that are there. You have to systematically number these parcels. You have to provide a serial number. You have to seal them and describe the nature of the seals. If you're giving alphabetical seals, you have to specify that I've sealed this parcel with 5 seals of suppose ABC. So that details has to be there. Also, what is inside that parcel and from where was that parcel collected. Right. So all these things need to be specified in that forwarding note, description, nature of examination. What do you want the FSL to do with the evidences that you are sending it? Suppose you are sending a blood vial which has been taken up from the victim and accused, and you are sending clothes of the victim. So what exactly do you want to do with this blood and this victim's clothes? So you have to specify that you want DNA profile to be created from both these blood samples, and you want to identify whether the semen is present on the female's clothes and whether that semen belongs to that particular suspect or not. So the DNA will be extracted from the clothes of the victim, and the blood, which is the reference sample of the accused. Both of these will be subjected to DNA extraction, and then ultimately the profile will be matched for the presence of semen. And if the profile matches, then you can report that these two belong to the same individual. If it does not match, then you will say that male DNA has been found, but it does not match with the DNA of the reference sample that has been sent by the investigating officer for matching. Right? What are the documents that are attached with these parcels that you're sending? That details also needs to be present on the forwarding note. And the signature of the forwarding authority and stamp of the forwarding authority needs to be present on all the pages, and the details of the messenger will also be present on the last page of the forwarding note. The constable XYZ with belt number 123 on date, suppose 6th of April 2024, is taking 5 parcels from such and such police stations, and he's authorized to bring it to FSL on the same date. So this is the complete forwarding note. What are the list of supporting documents that we have? In case of murder and homicides, we need autopsy specimen form, biological specimen authentication form, and attested copy of postmortem, and specimen seal of the medical officer, because in such cases, medical officer is sealing these particular exhibits. In case of assault or sodomy, sexual assault and sodomy, we need biological specimen authentication form, medical legal report, autopsy if the victim is dead, and there has to be a sexual assault victim information form that is going to give us details of the age of the victim, the time that has elapsed between the examination and the assault, whether the victim was sexually active, whether she was or he was below 18 years of age, whether he or she was married, whether he or she was having multiple sexual partners, whether she has any pregnancy. All these details are present on the sexual assault victim information form. And biological specimen authentication form is required in all those cases wherever you are taking biological samples from any living person, because you cannot take samples from a living person without his or her consent. Any point of time, the victim or accused can deny that they don't want to give their samples, because plucking of hair is painful. Injecting syringe for drawing blood is painful, so they can deny. But in high profile cases or in cases that are, grievous in nature, the court can order DNA examination, and in that case, both of them cannot deny. If the person is a minor, then she will the consent will be given by her parents. And in case of disputed paternity and maternity, the biological specimen authentication form, MLR and PMR, and a sample seal of medical examiner is needed. So in the laboratory, very, very quickly we'll cover that in the laboratory, how do we conduct the DNA examination? The first process is the extraction, then we do the quantification. After the quantification, we go into the amplification of DNA. Amplification of DNA is followed by separation or, a breaking of the double helix into single helix, and then analysis and interpretation on the genetic analyzer. So the cells are first broken and the nuclear DNA is released. Then, proteins and cellular debris is separated using proteinase K and other enzymes. And after that, the DNA is precipitated with alcohol and then it is separated. Then it is cleaned, purified. This is a very technical aspect, so I'm just giving a brief, rough idea, not going into the details of it. After cleaning the DNA, you have to confirm whether whatever liquid you have extracted, whether it contains any amount of DNA or not. So for this, previously we used to use a nanodrop for quantification of DNA, or presence of DNA per at the nano levels. And now we have real-time PCR, which helps us detect the amount of DNA per cycle. So PCR is cyclic multiplication of DNA. So RT-PCR is a technique where they help us to, look into the number of or the quantity of DNA that is increasing with every cycle. This is a graphical way of analysis. After this, you are amplifying and breaking the double-stranded DNA by the PCR technique. And after PCR, you're creating the DNA profile by using a 8 capillary or 24 capillary genetic analyzer, which uses capillary electrophoresis and creates an electropherogram, which creates the DNA profile of CODIS. Combined DNA index system right now is analyzing DNA on 26 plus 1 markers. The plus 1 is the amalogenin marker and 26 markers are there. So the first one, this is the extraction, automatic extraction machine. This is the PCR. This is the real-time PCR machine. This is the PCR machine. This is the nanodrop that we used to use, and now this is the 3500 genetic analyzer by applied biosystem. This is the new technique that is going to come up very soon. This is next generation sequencing or NGS, which is going to create profiles of nucleotide by nucleotide sequencing. Right now, we are just working on 26 markers, but in very near future, we are going to amplify and analyze each and every nucleotide sequence that is present in the human DNA, which is going to be more helpful in properly identifying the individual who is the offender. Where we have seen the importance of DNA evidence in India, I'll just discuss briefly 2-3 cases. This was, as we all know, the Kedarnath flood case in 2013, and the rivers that flooded, a lot of people died. And there were around 4,120 individuals from across the country that were reported missing. And the mortal remains of around 550 victims and blood samples of their relatives were matched at CDFD. And their unidentified or lost victims or lost remains could be matched with their family members, and then they could be identified who were dead and who were missing. The next very recent case was the Shraddha Walker murder case, where his boyfriend Aftab Amin Poonawala became friend with her on the Bumble app. And on May 18th, 2022, he strangulated Shraddha Walker and subsequently bought a saw and cut her body into pieces to destroy the evidences. He bought a new fridge on the very next day, stored the body pieces in the freezer so that there was no smell. And he spread these 17 pieces over different areas in Delhi so that they could not find her. But as per the 6,600 page charge sheet and his statements, there were remains of Shraddha were found at different places, especially at the Mehrauli and Chhatarpur forest area. And the remains of the bones and the hair, mitochondrial DNA of the hair, they were all analyzed and they matched Shraddha's father, and ultimately he was punished. There was Shiny Ahuja case, which was a negative case that I would want to discuss with you. Shiny Ahuja was convicted, were put in jail for 7 years for raping his house head. So what happened was on June 14, 2009, the girl or the maid who was supposed to be raped by Shiny Ahuja, she was called for investigation, and she denied any charges of rape or sexual activity from Shiny. She said that I had fallen in love with him, and on the day before I tried calling him a lot of times. He did not pick up the phone, and on the 14th of that day of June, a neighbor, women who saw her coming down just reported to the police that the maid was in not good condition. Her clothes were torn, her hair were disheveled. And another film person, Dr. Gupta, he took her to the police and made her sign a report which claimed that she has been sexually exploited by Shiny Ahuja. But in this case, there was no chain of custody maintained during the examination. No vaginal swab showed signs of DNA. Only there was one vaginal slide which was not subjected for examination of spermatozoa under microscope, and only this slide gave positive result for DNA. but there was no chain of custody maintained. Nobody knew from where these samples came, and they were not analyzed in a proper way. So initially, on improper examination report, he was jailed. And then when he filed an appeal, and when the investigating officer also said that I did not check the call records, that the woman was on phone at the time when Shiny was accused of raping her, and the CCTV footage also showed that the maid was coming down normally. There was no disheveling of hair, no torn clothes. So ultimately, he was released, and he was, freed of all the charges. Another case was Rajiv Gandhi case, where the bomber had a belt, and based on the remains that were found at the crime scene and the little amount of tissue that was present on the bomb belt of the body, these two things were compared for DNA profiling, and this was done by Dr. Lalji Singh only. And it was identified that his assassins were Dhanu, who was a woman, and Shivarasan, and it was conducted by Dr. Lalji Singh at Hyderabad. Right, and Sheena Bora, we all are aware of that. Initially, Indrani Mukherjee, she said that her daughter is in USA, but in 2012, her bones were found from Maharashtra area, and then they were tested with the mother and his father and her father, and then it was identified that it is the body of Sheena Bora. So that's it for the day. I hope this session was of some help for you. So thank you so much. So Jitsa coordinator can take over now. I'm done with my presentation. And if you have any query, you could also.
Thank you, ma'am.
Understand? Possibly.
Yes, sir. 1 minute, sir.
If anybody have any doubt, they can ask in the message section.
Shruti, any question in the comments box?
No, sir, no question. We can proceed for vote of thanks.
Okay. So first of all, Shruti, thank you for giving me opportunity to speak something about this session as a vote of time. First of all, I would like to brief this session within the short period of the time. we arrange this session, and when we decide with Mr. Nishne, Secretary of the NIFSA Craft, and with the HOD of the Department of the Forensic Science, we get a huge response from all over the India. We have got around 700 registrations from north to south, east to west, and all different kind of the field person like the advocate, forensic professional, student, faculty members, even some kind of the specialist person, even from the field of the medicine, from the from the doctors. They have filled this registration form, and about the today's content of this lecture by the resource person. So I personally would like to thank Ms. Pallavi for giving this kind of the lecture, and your content was very insight, very fruitful. You have covered entire area of the DNA from crime scene to the laboratory, which is very essential for packing, labeling, and forwarding also. Because many times we have seen that the forensic science laboratory did not accept the case because of the correct packing, and sometimes we fail to analyze the DNA for the positive identification because of the improper collection. So this is very important for all the participants. Now I would like to go for the vote of the thank. So first I would like to thank Ms. Pallavi for giving your time, for your contribution for this session. Another, I would like to take name of our director, NIFSA, for giving me opportunity to arrange this kind of the session for the all kind of the participants. The next person, that is the Dr. Rajneesh Kumar, HOD of the Department of the Forensic Science, with discussion and without the HOD and without his efforts and time, I cannot proceed this kind of the event. So I am very thankful for him. Now I would like to take the next name, that is the NIFSA Craft. All our student, our NIFSA, they utilize their time, they utilize their efforts, and they arrangement this events. Especially, I would like to thank Mr. Nishne, Shruti, Arya, and Mahesh, and all other the members of the NIFSA Crop. With this, in the last, I would like to thank all the participants who have registered for this kind of the session and who listen or who join this kind of the this meeting for this session. So all of you, all the persons are very thankful for joining this kind of the meeting. Okay. So now I would like to hand over mic to Shruti for ending the session. Thank you.
Thank you, sir. I request all the participants to please fill the feedback form. After filling the feedback form, only the certificates will be provided.